pleased to welcome Joel Richard. Joel is the Senior Vice President for Peptides at IPSEN. He is located in France, where he's responsible for global pharmaceutical development of small molecules and peptide-based products. He has over 25 years of experience in chemistry and biopharmaceutical R&D. Joel obtained his PhD in materials sciences at the University of Paris in 1987. He was the founder and COO of MainLab, a drug delivery company which specialized in developing solvent-free processes for proteins. After that, he was vice president of research and R&D at Ethypharm in France, and then director of pharmaceutical development at Serono and Merck Serono in Italy and Germany. Since 1996, Joel has focused his research activity on new formulation technologies and drug delivery systems with a focus on injectable peptide and protein formulations. Joel, all yours.
So thanks a lot, Mr. Chairman, for your kind introduction. And I would also like to thank the organizing committee to invite me for this conference. I'm very happy and very proud also to be here because I know this is a top-level conference in the field of peptides. So I'm working with IPSEN. So IPSEN is highly involved in peptide development, as you know, and commercialization. We have more than 8 different drug product forms on the market, from immediate release to sustained release formulation until 6 months, using various technologies. So, this assembly is well expert in the field of peptides, so it makes my task quite easy, and I'm not going to talk too much about how difficult it is to administer peptides because of their inherent properties, physicochemical and biophysical properties, such as short half-lives, sensitivity to degradation, enzymatic degradation, pH, and so on. So today, I would say that most of the peptides that are systemically active, bioactive, are administered through injection. And there's a tremendous interest today in alternative routes of administration of peptides. I would say less invasive routes of administration. And in this context, this is, I would say, the purpose of my talk, to review the non-invasive routes of administration of peptides and then to focus on the oral administration. So let's have a look at the peptide delivery landscape and the importance already of non-invasive routes of administration. So, as already mentioned, most of the peptides, and more particularly the ones that are systemically bioactive, I mean the ones that are not targeting receptors in the GI tract, they are administered through injection. But as you can see here on this slide, I would say there are also already quite significant routes, other routes that are used, such as the nasal, for instance, but also the transdermal one that are considered and potentially used. So if we look at the technology landscape now, you can see on this slide that, most of the injectable peptides today are sustained release formulations. It's a kind of standard of care from 1 month to 6 months, using most of the time PLGA microspheres or implants. There's maybe one exception, and I'm quite proud to talk about it. This is the somatolene autogel, known as somatolene depot formulation here in the US, which is commercialized by IPSEN for the treatment of acromegaly and neuroendocrine tumors, which is based on self-assembly, peptide self-assembly, which means a very simple formulation containing only peptides, water, and a buffer. But let's come back to the presentation here. So, so you can see that, these are sustained release formulations. Sustained release, why? Because you want to, I would say, limit the number of injections. And of course, if you can have one injection every 6 months instead of one every day, it makes a big difference in terms of acceptance of the treatment. And so this is key. And second aspect I would like to mention is that there are also some nasal formulations that are on the market, that have been on the market for a relatively long time. But for this type of formulation, it is not recommended to use them on a very long time and for chronic administration. And also, their efficacy depends strongly on the adherence to the treatment. So if you miss one administration, you may be out of drug and out of efficacy of a drug for the time you miss your treatment. So non-invasive delivery routes of administration, what are we talking about? Here they are almost all presented, I would say. It goes from pulmonary, nasal, transdermal, transmucosal, and oral. And you can see here, I'm not going to go into all the details. The pulmonary story is well known with some, I would say, stories about Exubera or Fraser for insulin, not so successful from a commercial point of view. Nasal also is presented here, and more than 20 years on the market, but maybe not successful neither, with high variability and potential issues with treatment adherence. Transdermal is progressing fast, and more particularly we will see with the microneedle technology. As regards the transmucosal and more particularly the buccal, it seems to be very attractive, and there are some interesting studies in the field of oral insulin, buccal insulin, sorry, with the oral product, spray product commercialized in some countries by generics. And finally, the oral one, which is progressing very fast. And I'm talking here about peptides that should be that should cross the epithelial membrane. I'm not talking about peptides having receptors in the GI tract, and I'm, for instance, talking about oral GLP-1 analog or oral somatostatin analogs. We will come back on this. So if we just have a look at, before going to the oral, what about the transdermal? So transdermal, as you know, a peptide or a protein will not cross the skin by itself because it's too big and too hydrophilic. So you have to help it to cross, and one way is to create transient micropores, microchannels, and you have different possibilities to do it, either pulse currents, laser beam, alternative currents, or microneedle array. And actually, this is something that has been progressed by many different companies, as you can see here. And well, I would say that some of them have already stopped, but well, you know, this is something that may come back. You never know with technologies, and this is the beauty of our science and technology. And I would like to focus more on the microneedle technology, because actually if you consider solid microneedle arrays coated with peptides, like for instance the one from 3M, well, it was published with a PTH analog, which by the way was developed by IPSEN a few years ago and licensed to Radius, that well, you can get very interesting fast onset of action and also PK with this product. And another development is based on hollow microneedle array. So in this case, these are microneedles where the peptide solution can be administered, and you know, we like to have a cartridge with a solution of peptides because this is something we know very well, and that could be adapted, for instance, in this type of system and be administered through transdermal. And it was shown that the PK is quite close to the one that can be obtained through subcutaneous. This was not the case, by the way, for the subcutaneous peptide here. You can see the transdermal peptide is much faster onset of action than the subcutaneous one. But I would say that the area under the curve was interesting also for the transdermal one. So this is very, very promising. And I would say that there are a lot of advantages related to the fact that this is non-invasive. It avoids the GI tract, as you know. So no exposure to media where the peptide can be degraded. This is also expected to show higher bioavailability versus the oral administration, and we will have a look at it. But there's a but. What about immunogenicity? Because actually you are exactly administering the peptide where the immune system is sitting and waiting for everything which is foreign to process it and then to train the immune system against this what you have administered. So, well, this is something still to be solved. As regards buccal delivery, so buccal delivery, it means you have to go from here to there. So it seems to be quite a long way with a lot of and complex, I would say, environment to go through. But drug can be absorbed by permeation across the buccal mucosa, and most particularly, or more probably, let's say, by the paracellular transport. And actually, this is something that was shown a few years ago with oral lean, so it administered buccally. This is a liquid micellar formulation of insulin administered through an aerosol with a specific device. And so actually it has a lot of advantages compared to other systemic insulins, such as, for instance, the fast onset of action. And also I think it's ideal for prandial insulin to be administered immediately prior to the meals. It has also a lot of advantages because it does not need to go through the GI tract, so you are not expecting high degradation of a peptide. It's not exposed to harsh media, and so the bioavailability is expected to be also to be higher. In terms of patient compliance, this is also something that, for instance, compared to other alternative routes that were proposed, like the pulmonary one, with maybe, I would say, devices that were maybe not so adjusted, that may be a quite interesting alternative. And the fast onset of action is also something that is expected from the patients, you know, so it's quite a big advantage. It has also limitations because, the buccal environment, you know, it's constantly renewed, and which means that you may have to develop a mucoadhesive system to avoid that the formulation is removed too rapidly from the mouth. There might be also some acceptability and taste issues, and on the long term, you may also expect some damage of a buccal wall. And finally, the absorption can be also limited by various factors, and depending on, I would say, your also health state, your personal health state. Okay, so it was just to give you an overview about, I would say, two very promising non-invasive routes of administration, which are the transdermal with the microneedles and the buccal, most likely, which are still to be more investigated in terms of peptide delivery. So, as a summary, if we look at peptide delivery and the technology's trends, I would say that injectable sustained release will remain the key one, with improvements related to ready-to-use formulations in pre-filled syringes. Today, this is not something which is easy to achieve for sustained release formulation, to have it ready to use in a pre-filled syringe. So you know also another important thing is that we have to focus on narrow gauge needles, but as you know, if you look for microspheres, it's quite difficult to have narrow gauge needles with microspheres. So other technologies have to be found, and why not, I would say, peptide self-assembly. Transdermal peptide delivery is a technology that is progressing fast into phase 2, and I'm quite supportive of the microneedle technologies. Oral peptide technology, we are going to focus on it, and there are a broad range of technologies for systemic absorption that we are going to review. And finally, I would also like to mention, even if I'm not going to talk about it, that there's another important area, which is the intracellular peptide delivery, targeting cancer cells, and more particularly to modulate protein-protein interactions. Another, I would say, interest of intracellular peptide delivery is also the crossing of the BBB. So now, let's focus on oral peptide delivery. So first point I would like to mention is that there are strong expectations and also large opportunities. Strong expectations because if you ask, for instance, somebody who is on a somatostatin analog treatment, so injectable products, whether they would like an oral one, and I would say that more than 2/3 of these people, they say, yes, we would like it. So there's a strong expectation, which is also mentioned here by Casma when they developed their oral octreotide. The idea being that, we could provide an option to patients to avoid painful injections. Opportunities, there are opportunities for delivery, for instance, to a site of action which is in the GI tract, and actually there are products that are developed and commercialized today. There's linaclotide that recently there was another one, Trulance, for instance, that was put on the market. And another opportunity is to deliver to the liver, and this is maybe one of the reasons why so many people are concentrating on insulin or GLP-1 through oral administration, because it makes sense as you are. These products are supposed to be active in the liver, that you are trying the oral route to make them target the liver. Okay, so I would say that, this is also a very competitive environment, and there are a lot of hurdles. As you can see, Casma had, for instance, a first partnership with Roche. Then they got back the product. They developed by themselves. It's a very courageous way. They filed the product. FDA refused first, but they insisted, and now they are going for a new phase 3 with a protocol which is approved by FDA. So I would say, this is a long way, but very strong people that want to develop these products. And as regards the GLP-1, Novo Nordisk is a front runner in the field, investing a lot in this area with different technologies, including the Hemisphere 1. So I will not insist on this one. I think it was a very interesting one. It's targeting receptors in the GI tract with linaclotide. There's a cascade of events with two effects, basically. One, secreting fluid in the intestinal lumen and accelerating the transit. And another one having a systemic effect on pain receptors, which control actually the pain of these patients with inflammatory bowel syndrome. So if we talk about marketed oral peptide today, here is a list. I do not pretend to be exhaustive, but you can see that most of these peptides have a local action in the gastrointestinal tract. They are targeting receptors in the GI tract. This is the first observation. Second one is that most of them are cyclic peptides. Well, most likely because they resist more the degradation from exopeptidase. There are, well, I would say two of them that are anyway absorbed by the system, which is good news because it demonstrates the viability and efficacy of these products, even if the bioavailability will remain very low, a few %. So the main challenge, as you know, this is how to cross the membrane, the epithelial membrane, and there are different pathways. Either the paracellular one, which means that the peptide will go through the epithelial cells, and you may have an excipient helping opening these tight junctions. There's the transcellular one, which means that you have to go through the epithelial cells, and once again, the peptide may be helped by using an excipient that will form a physical complex with a peptide and make it more hydrophobic, or let's say more amphiphilic, and so able to cross the membrane. And so, as already mentioned, there's an opportunity to target receptors in the GI tract for local treatment or to trigger a signal transduction cascade, but there's also an opportunity with novel excipients or nanoparticles approaches to cross the membrane. But if you look at the ratio of the surface of the cells versus the surface of the tight junctions, you can see that this is a quite unbalanced situation, and you may imagine that the probability of crossing most likely is higher through the cells than through the tight junctions, just for a physical reason. So, here is a summary of formulation excipients that are used in key oral peptide formulations. Why do we use these excipients? The first one is that there's a need for stabilization. And stabilization is first stabilization against enzymatic degradation. This is why people use enzyme inhibitors, which are quoted here. There's also a need to protect against pH. It's not mentioned here because it's quite easy technology using enteric coating formulations. You protect from degradation in the stomach. And most importantly is the permeation enhancer, because if you don't help the peptide cross the membrane, the bioavailability will be very, very low. And even with these technologies, you will stay in the 1% range, okay? So here, these are different permeation enhancers, so they help the peptide to cross the membrane. Some of them are designed to go through the cells. So this is the transcellular pathway. Some of the ones, like the fatty acids derivatives, caprylic acid, are known to be tight junction openers. So I would say, this is today the most commonly investigated and successful approach for peptide absorption. As regards the enzyme inhibitors, I would like to mention that one point that needs to be carefully managed is the safety profile, because actually, the chronic use of these enzyme inhibitors may have an impact also on the GI tract. And more prospective approach is based on functionalized nanoparticles, and I will come back later on this aspect. As regards mucoadhesive systems, some people use mucoadhesive systems. The question is, how do we manage the interactions with the mucus? Is it good to have I would say, retention at the mucus level, or conversely to try to cut the mucus to access in an easy way to the epithelial membrane? So it's not yet clarified. So here is, I would say, one of the first technologies that were developed in this field. So this is the peptiligence technology from Antares Biopharma, formerly Unigerm. And you can find in this formulation, I would say, all the main components of formulations for oral peptide delivery. The permeation enhancer, for sure. You have also some pH control systems also to avoid the action of enzymes. You have also, I would say, enteric coating for the peptide to survive in the stomach, and of course the API. And this is the first technology that went to phase 3 for oral calcitonin. This is why I think it's worth mentioning it. And then it has been developed for other indications and other type of peptides. But today, well, I would say it's still something. So you can see here it's a press release by January 17. It's still in the radar of big pharmas for oral delivery of peptides. Another one is the one from Merion Technology, and this one is based on tight junction openers, the sodium caprate and caprylate system. And actually, this was initially developed for insulin and oral GLP-1 administration. And by the way, Novo acquired it. And they evaluated it in clinical trials, but finally they dropped it, they stopped, and they focused on another one, which is the Hemisphere one, with the Eligen product, with the famous, for instance, snack excipient. And actually GLP-1 now is going into phase 3 with the Eligen technology from Hemisphere and no longer with the Merion technology. So here, these are the peptide technologies progress in the clinic. So you can see that, there are a lot of studies are focused on GLP-1 and insulin. And you can also see the octreotide product from Chiasma, which is now in phase 3. And you can see that, they almost all use permeation enhancers, different ones, because actually these are kind of proprietary technologies. And also some of them use nanoparticles. And I think it's quite interesting to see that different approaches are used for these clinical studies. But which means that oral technology of peptides is something which is now progressing in late-stage clinical trials. What do we learn from these clinical studies? First, that with an oral peptide, you can achieve similar pharmacokinetic compared to subcutaneous injection. This is a good news. But the dose is very different because bioavailability is low, which means that, for instance, 20 milligram octreotide is equivalent to 0.1 milligram 20 milligram oral octreotide is equivalent to 0.1 subcutaneous octreotide. And in addition, there might be effects related to food administration and interactions with drugs like proton pump inhibitors. The other good news is that the peptide which is administered remains bioactive. And this is shown here. Once again, this is with oral octreotide by the team from Chiasma. And they showed that with the oral octreotide, you control well the GH level even after the test. If you have a challenge with GHRH arginine, you can see that there's a good control, good bioactivity of the oral octreotide, which is good news. In this case, you can also find that you have a higher efficacy for the oral peptide delivery versus the reference treatment. Here, the reference treatment was the nasal formulation of salmon calcitonin, and versus the nasal formulation, the oral formulation of calcitonin gave a better efficacy, which is also another good news. So you can see that there are a lot of good news. However, the product that was submitted in 2015 has not been approved by FDA yet. So, well, I don't know what's going on. There are discussions, obviously. Well, another thing I would like to mention, and this is a point that will need to be addressed for further development of oral peptides. This is about food effects. So it was shown for the chiasma technology, but this is not specific of this technology. This is the purpose of this slide, to show this. This is another technology. This is the hemisphere technology for oral calcitonin, and they showed that depending on the amount of water that you take with the oral formulation of peptide, and also depending on the time before the meal, you have different absorption, which means that you will need to have a very strict regimen for administration in terms of what do you take with the drug, and also how much time before the meal, and so on. So it brings us rapidly to the future challenges and prospects. So if we want to switch from injection to oral route anyway, we have to consider that the relative bioavailability will remain very low, which means that, well, question is whether there's a real interest versus, I would say, 3 months or 6 months injection. In addition, what will be the market access? Will we be able to demonstrate superiority versus reference treatment, and also better safety or efficacy? And there's a big impact on cost of goods, because actually if bioavailability is low, it means that you have to produce, I don't know, 20 times or 50 times, 100 times more, which means that you have to invest. And actually, this is what Novo Nordisk is doing now. They published this in March 2017, where they say that to bring GLP-1 to the oral phase 3, they invest how much? 2 billion. Wow. $2 billion for manufacturing sites in Denmark and North Carolina. So, well, I mean, here is the proof that you need to do something in your API manufacturing to go to oral peptide. And now, if we compare oral bioavailability versus competing technologies, you can see that this remains very low compared to all the other ones. And the question is how to further improve it. And this is now the perspectives. So what are the perspectives? And, well, this is still a very prospective approach. But I'm really thinking that targeting nanoparticles, for instance, here, this is shown for FC functionalized nanoparticles that have I would say, a very specific interaction with the neonatal FC receptor. So here is shown a pathway for transcytosis transport through the epithelial cells. So here, these are nanoparticles that are made of block copolymers, PLA, PEG. So you can see that you get very nice nanoparticles, well functionalized. Size is in the 50 nanometers, and in vitro, well, they are convincing us that they can they can cross in an easier way the membrane compared to the known functionalized nanoparticles. So, well, very interesting approach here, based on the targeting to the surface, and also on the fact that the nanoparticle can be loaded with a peptide. And here you have also the proof that absorption is better in animals. And, so it's clear here that in the mice administered with the FC functionalized nanoparticles, the absorption is much better compared to the non-functionalized one. Distribution also is different, as expected, I would say. And there are other systems now that are coming. For instance, nanoparticles that are functionalized with vitamin B12 analogs. And these people, they showed, so this is the Coboral technology from Access. They showed insulin uptake up to 29% versus subcutaneous, which is very good. I would say. So, well, I know these are only mice, this is not human, but it's very promising. And here is another approach, still based on nano objects. These are nanocapsules, and these are CPP functionalized nanocapsules for oral delivery. This was published very recently in September of this year. And you can see that these people, they form nanocapsules with polyarginine, and they showed a good stability in the GI fluids. They also showed that, as you can see, so it's related to the guanidine groups and the CPP property, and stability is shown here in the GI fluids. And what is also very interesting is that actually they loaded it with an anti-cancer peptide drug, and with a molecular weight in the 1,600 gram per mole, and they showed that it has a low cytotoxicity in CACO2 cells. So I think it's also a very promising aspect, because you may expect that these highly cationic groups can be highly cytotoxic also. And it was also demonstrated that loaded with a fluorescent molecule, there was an enhanced gastrointestinal retention, which I think is also very promising in terms of activity. So these are preliminary results. They show also that potentially these polyarginine nanocapsules could favor the paracellular transport of hydrophilic drugs, and so could be, I would say, the next generation of oral peptide delivery systems. And before going to conclusion, another one, which is quite different, but also very interesting on the concept. It's a really innovative. This is a intra-enteral injection. So I know that some people would say, oh, I don't want to have an injection in my intestine. And I understand the point. But so here there are two systems actually. One is based on an inflatable balloon at the surface of which there are some needles made of sugar that can be broken off, and then they are loaded with a drug, and they release the drug in the intestine wall. And another one is made of metal microneedles. So this one is from the MIT in Cambridge, and this one is from Rani. And actually, this is attracting a lot of interest by big pharmas for biologics, for peptides, and you can see that I would say a lot of companies, Novartis, AstraZeneca, Novo Nordisk, are attracted by this system for oral peptide delivery. And of course, you are expecting a much higher bioavailability with this system. So let's come to the conclusion. Rapidly, a summary. Key points are peptide stabilization versus enzymes versus pH. Bioavailability will remain very low versus other routes of administration in the 1% range. Variability very high. We have to also focus on the absorption enhancer safety, because actually they should be only transiently opening the tight junction, for instance, or favoring absorption. Food effects and drug-drug interactions are to be taken into account. And anyway, well, proven efficacy is here versus injectable sustained release formulations. And Hemisphere, I think, is now going with Novo Nordisk for GLP-1 analog in phase 3, which is, I think, a big step. So, well, I think I will not go through all the details of the conclusion. Oral formulations of peptides are highly desirable. Proof of technology has been made. Chiasma is now going in phase 3 with an approved protocol by FDA for somatostatin analog by oral route. And in the next years, injectable remain the main route. Oral products will come to the market. You have seen you have already about 10, so there will be between maybe 10 and 20 in the next 5 to 10 years. And key point is about bioavailability and the innovative approaches that are proposed for improving this bioavailability are targeted nanoparticles and intraoral microinjection. Now let's see what the next products on the market will give. There has been I would say, not so good experience, for instance, with pulmonary insulin, first products coming to the market, then being retrieved and killing all the other developments. Let's hope that it will not be the same for the oral peptides. This is just as a conclusion. The way to give is at least as important as what is given. This was a saying by a very famous French theater author in the 17th century. I think it's very true for everything and for drugs, and this is why we are here. And so I would like to acknowledge co-workers' contribution, Leila Hassani and Andy Lewis, and I'm happy to answer questions. Here is literature which is related to this work. Thank you very much.